Mhm.
All right, it's about that time. Hello, everyone. Thank you for joining us for Clarice's speech today on quantum biology. I know we're all really excited to hear all that she has to say. And this little, this thing is so cute. I don't even want to call it a comic strip. I love it. It's awesome. But I will hand it over to Clarice. And unfortunately, I will be turning off the chat, but I will be turning it on in between lecture and the talk before we get started. All right, let's begin.
I am so happy to be here. Hello, everyone. And I'm going to say again on the record, I truly believe that qubit by qubit is the best thing ever to have happened to quantum education. You are so fortunate to be able to learn with them. I am so proud of their work. So congratulations on your being here, on your putting effort into learning quantum computing. I'm from Brazil originally. That's where the accent that you're hearing, and my pronouns are she, her. But I like to call myself a quantum engineer. This means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics as opposed to the laws of classical mechanics that rule everything around us. So people get a little bit surprised when I tell them that I study things that might be going on inside birds, inside butterflies. So before I explain where I'm going, I really need to tell you where I come from. We're going to start the talk with hardcore quantum mechanics, in particular, something that people call quantum sensing. And by the end of the 30 minutes, we will have talked about things of biological relevance, such as organismal migration, how our cells sense oxidative stress, how our cells react to radiation. And I hope I will have convinced you that I think we can learn with nature on how to build better technologies. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. Usually, I start my talks by saying that I think humankind is sort of obsessed with measuring things better because this might mean that we understand things better. So we can think about measuring better frequencies. This big machine is a atomic clock that sits at NIST, the National Institute of Standards and Technologies in the US. And this machine helps us define very well what a second or what a hertz is by using atomic transitions. We can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. Or we can think about measuring better accelerations so that our gaming experience is enhanced. Those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very small? Or worse, what happens if the object causing the quantity that you want to measure is very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. For reasons that I won't have time to fully explain, you can actually mathematically prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. In my past, I worked with a technological quantum sensor. And I'm going to take some time to describe it to you because I'm going to use it as a comparison point to another type of a quantum sensor, this one made naturally by nature. Okay, so let me talk to you about the technological quantum sensor that I worked with in the past. This quantum sensor, in particular, was an electronic spin in the material diamond that was put to use as a quantum enhanced magnetometer, magnetic field sensor. So, for those of you that have never heard of spin, you're in for a treat because spins are super cool. So, spin is a fundamental property of matter that is as fundamental as matter and charge. Okay, in the same way, and things have spin, like small things have spins. For example, atomic nuclei have spin, electrons have spins, and in the same way that, for example, mass is the quantity that reacts, that interacts with gravitational fields. Charge is the property that interacts with electric fields. Spin is the quantum property that tells us how objects interact with magnetic fields. And in the end, it's all a big abstraction. In the same way that you have negatively charged objects and positively charged objects, right? For example, negatively charged objects are electron, positively charged objects are like atomic the protons, right? In the same way that for the electric field, we use positive or negative to denote two different interactions with the electric field. In the case of spin, we usually denote the different ways of, say, an electron interacting with magnetic fields by saying that an electron can either have spin up or spin down. So spin is actually drawn by chemists, physicists, engineers as an arrow. Arrow pointing up is usually referred to as spin up. Arrow pointing down is usually referred to as spin down. And spin up or spin down just refer to different energies of say an electron interacting with a magnetic field. Okay? So here's the promise that I'm going to explain to you how to put a single electronic spin in diamond to work as a quantum enhanced magnetic field sensor. So here's how this works. In fact, the spin that we can play with in the material diamond, again, it's absolutely at room temperature. There's nothing special. The material diamond is actually the material that some of you might have seen in jewelry, right? The electronic spin that we can play with to put to work as a magnetic field sensor is actually comes from a trapped electron in this material diamond. So there's actually a lattice defect in the crystalline matrix of the diamond, and electron gets trapped, and we can use this electron spin as a quantum sensor in a way that I'm going to describe. Okay. In particular, and I'm mentioning this only because I'm going to mention something else later on that I wanted to compare with. This particular electron spin is associated, this defect that traps this electron is associated with a very important property that it absorbs light. Okay, so this means that if you shine light onto such a crystalline defect that traps an electron in a material diamond, this defect is going to absorb light. Upon light absorption, the defect is going to get excited, and as it de-excites, it emits light. Okay, so it fluoresces, it emits light. So it turns out that, again, for reasons that I won't have time to explain, this defect in diamond emits a different intensity of light if that particular spin is up or down. This means that just by looking at how strongly the defect is emitting light upon excitation, you can actually do a readout of the quantum state of the electron spin up or down. In jargon, and at some point you will be introduced to this language, this means that this defect has a quantum state dependent fluorescence intensity. Remember this. So this is how you do state readout of the defect, just by looking at different fluorescence intensities. But that's the promise that we can put to use this trapped electron whose defect fluoresces to work as a very sensitive magnetometer. And first of all, I put in this to the right here in this scheme the two different spin energy levels. So the idea is that in the y-axis you have energy levels, right? And you have two energy levels that are marked there 0 and 1. You can think of one representing spin up, one representing spin down, and their difference in energy is in the microwave range, more or less the range of the waves inside your kitchen's microwave should you have one. Okay. And it turns out that from those two states, one of them, the one marked with 0, is insensitive to magnetic fields. This means that if you immerse the diamond with this defect into a magnetic field, state 0 will not care about this magnetic field at all. However, the state marked +1 is sensitive to magnetic fields. This means that if you put the diamond material in if you approach a magnet or something, or if you immerse this magnetic field, this diamond in a magnetic field, state one will actually be perturbed. It actually will be promoted in energy, or have a shift in an energy by a tiny quantity that I mark delta there in my picture. And if you're still sleeping, this is the time to wake up, because the point that I'm going to make now is extremely important. It doesn't always get made. And this is basically what underlies about 90% of the modalities of quantum sensing. The idea is that this tiny shift in energy that the state marked with one senses is actually proportional to the magnitude of the magnetic field that the diamond is immersed in. So that actually the problem of measuring a magnetic field is actually mapped onto the problem of the experimentalist knowing how to measure a mismatch delta from a known energy difference, right? The energy difference in the absence of magnetic fields. And this again is the idea behind many quantum sensing modalities. You have a certain quantity that can be magnetic field, electric field, temperature, something else, and you know how this particular quantity shifts a known energy difference, right, according to some law. So if you know how to measure such shifts from resonance, by extension, you know what the quantity that caused such a shift was. So and it's fundamentally a signal processing problem. And happily, engineers and physicists have known how to measure mismatches or energy shifts from a known resonance. They have known how to measure it for many, many decades. Okay, so this is how we can put this spin, or this spin in diamond, electron spin in diamond, to work as a magnetic field sensor, because we know how it's the two energies, the energy difference between spin up or spin down, how they change in the presence of a magnetic field. And we know how to measure this change in energy. So the big idea is, well, you have a tiny little sensor, right? Maybe a tiny little nano diamond that is, say, 20 or 15 nanometers in size, that contains one or many of those crystalline defects that you want to study. So it's a tiny sample, tiny sensor that can get very, very close to a tiny sample. And because the experimentalist knows how to measure shifts in the energy levels of those spins, you actually can measure magnetic fields produced by the samples. And actually, there are already many practical applications of such sensors. There are people using those sensors that work well at room temperature, and in the messy conditions of the proximity of a biological sample, say the messy conditions of a solid state lattice. So people have been using such a defect to make better magnetic resonance machines. People are using these sensors, these quantum sensors, to prospect for oil. So those are technological quantum sensors that use a very unique quantum property to work, and actually can be proven that they work better than classical sensors. What are your questions for the time being right now? I see
Uh,
some yes.
oh, you see them? Okay, perfect.
Awesome. So, is spin up one? I am not going to answer. Well, I'm going to answer this question, but it's just as a parenthesis. Okay, most electrons, probably all the electron spins that you're going to deal with in your qubit by qubit class, they are spin one half. This means that they can have two uh, states, arrow up or arrow down. Okay. It turns out that for the spinning diamond, it has an effective spin one, not one half. This means that it has three possible quantum states. It has, like, if you will, it has +1, 0, and -1. So it has three quantum states. But we usually want to deal with a qubit, right? So in fact, we single out two of those three available energy states and make a pseudo qubit out of it. So, for example, we take the state 0 and 1 as our effective spin states. So it's not really that spin one and zero, they do not correspond to effectively up and down. Okay, so but it's rather a spin that is created out of two energy states out of three available. But this is just a parenthesis. If you didn't understand anything that I said, don't worry. This is really just a tiny footnote for you right now. Um, so how does the electrons in the diamond move? So the cool thing about this defect in diamond is that it does not move. Okay, that's the important thing because it actually is trapped there. So once it's there, it doesn't go anywhere. You can keep playing with this more and more, right? Um, what kind of information do we gain from the measurement in relation to a biological setting? Well, we can measure, for example, magnetic fields produced by things inside your sample. We can measure temperature of cells. Already people shoot those diamonds inside cells and they can measure differences in temperature, right? Um, what more precisely is the defect? This defect, and you can Google it, is called the nitrogen vacancy center. It actually happens naturally in diamond. Actually, the first sample that we ever worked with was a discard from a jeweler, but people can also engineer this. This defect happens when a vacancy, that is a missing carbon atom, sits nearby to a nitrogen, that is the most commonly occurring substitutional impurity in the diamond lattice, which is only composed of carbons. And with this, I'm going to go to the other part of my talk, but I just want to make sure that I mentioned that I've just presented you with a quantum sensor that works at room temperature and under messy conditions. Okay, so and just up to 5 years ago, I was used to dealing with this type of sensor that you see there, the sensor that I just described, those big technological sensors. But at some point, I realized that nature many times um, produced sensors that in crazy ways outperformed humankind made sensors. Okay, and I'm going to describe to you a magnetic sensor that seems to be working in a way that is very similar to the technological magnetic field sensor that I just showed to you. Okay, so this is how I'm going to make the connection between quantum and putatively biology. But if we want to talk about biology at the nanoscale, we better start with biology at the nanoscale, which is actually the chemistry, right? So it's been known from test tube chemistry, no doubt, this is a known field, that magnetic fields can alter the final products of a class of chemical reactions that depend on spin. Now you should be looking at me. So here's the idea. The idea, there's no controversy in that. The idea is that there's a class of chemical reactions that the chemical reaction is happening, and at some point the chemical reaction comes to a crossroads. At that point, the chemical reaction effectively looks for a certain electron spin state. If the spin state is found to be up, I'm going to use a jargon, and maybe in some months you can come back to this lecture and see if you understand this jargon. If the electron spin is measured by the environment, if you don't understand this jargon, doesn't matter. If the electron spin is up, the chemical reaction continues through one branch. If that particular electron spin is down, the chemical reaction continues through another branch. Importantly, the final products of those two branches are macroscopically different. Okay, so a finicky quantum property of matter actually have been shown to macroscopically change, like big time, the output of chemical reactions. Now, if at the moment when the chemical reactions is at this crossroads, the electron spin of interest sees interacts with the magnetic field, a phenomenon that is indistinguishable from the type of quantum sensing of a magnetic field that I just described actually takes place. So this electron sees the magnetic field, and for reasons that I won't have time to explain, but reasons which are absolutely indistinguishable from like how that electron in diamond interacted with magnetic fields, the magnetic field might actually alter the probability of us finding the spin up or the spin down in such a way that this magnetic field might actually alter the probability of the chemical reaction continues through this branch or that branch, thereby also being able to alter macroscopically the final products of the chemical reaction and at much, much longer time scales. Whereas this interaction with the magnetic field is very, very brief, everything happens downstream and at much, much longer time scales. This has been demonstrated at room temperature, in solution, in the gas phase, in the solid phase, and for magnetic fields, down to the strength of the magnetic field of the Earth, which is weaker than the magnetic field that you sense from your cell phone when you put it like this. So this whole conversation migrates from chemistry to biology because of birds. Okay, I don't care about birds, but birds have the very important advantage that they brought this conversation to chemistry and to biology and to quantum physics, if you will. So birds, when they migrate, they know, again, no controversy in that, they know to be able, they know to use the magnetic field of the Earth to migrate, at least as a partial cue, okay, when they do their migrations from south to north during the winter to summer or something. And again, the magnetic field of the Earth is tiny, tiny, like way smaller than the magnetic field of your cell phone. How might they be doing this? So in 78, a brave biophysicist named Klaus Schulten, he made a crazy at that point hypothesis. Here what he proposed. Nobody took him seriously. He said, well, were the same type of spin-dependent chemical reaction happening inside the birds somewhere, birds and other organisms in general might sense magnetic fields to the extent that they might sense the different physiological products coming from those two different branches of those spin-dependent chemical reactions that depend on magnetic fields. That was a hypothesis. And some people hypothesized that maybe there was some sort of like a bird turning right or left would interact with different magnetic field lines, and maybe the different outputs in the chemical reactions would be maybe a modulation of the eye sensitivity of the birds so that the birds would see brighter or darker depending on where they looked, and in this way they could see the magnetic field. Now this has never been either proven or disproven for birds, but experimental biophysicists started looking for molecules that could actually sustain spin-dependent chemical reactions. And at that point, the only animal because of birds, the only animal protein that was known to sustain spin-dependent chemical reactions was this protein called cryptochrome. Now, cryptochrome is a fluorescent protein. That is, it has a fluorophore. That is, if you shine light at it, it gets excited and then fluoresces, pretty much like that defecting diamond that I just described to you. And birds have cryptochromes in their eyes, migrating butterflies have cryptochromes in their antennas, and actually us, humans, we have cryptochromes in all our cells because cryptochromes also have circadian rhythm regulation functions in all our cells. So it turns out that over those past more than 40 years now, there is a lot of evidence correlating, macroscopically correlating cryptochromes to magnetic field sensitivity. And cryptochrome is also present throughout the tree of life. All those organisms there, they have cryptochromes in their cells, and for many of those, there are macroscopic experiments that somehow relate cryptochrome to magnetic field sensing. And again, evidence for this phenomenon that cryptochrome actually mediates the sensing of magnetic fields via spin-dependent chemical reactions, the evidence for that exists, but unfortunately at very disconnected length scales. For example, to the left of my slide, you will see the macroscopic length scale. So for cryptochromes in solution, there is zero doubt that cryptochromes and the spins in cryptochrome are behaving as bona fide quantum sensors. For example, and that's the top left plot there that you see. Researchers, they took a vial of cryptochrome, shone a laser onto this cryptochrome, okay, and they tracked the fluorescence of this cryptochrome as a function of the time during which you shone this laser. The first thing that you see is that this fluorescence is going down. Basically, this means that the laser is killing the protein, is messing up with the molecular structure. This is called bleaching. But as the researchers, as this fluorescence was dying out, as the researchers post a tiny magnetic field on and off, they also see, and as you see in the inset, they also saw a modulation of the intensity emitted by this cryptochrome. Now, if you remember, I told you that for the spinning diamond, just by looking at how strongly it was emitting fluorescence, you could infer if the spin was up or down. Here is actually the same thing. just by looking at how strongly cryptochrome is actually emitting light as a function of magnetic field, you can infer if the spin was up and the chemical reaction continued through one branch, or if the spin was down and the chemical reaction continued through another branch. So for molecules in solution, this is a bona fide quantum sensor, even though the chemistry community is still not using this language. However, the next step in the wave in the length scales happens for big organisms, for example, birds during migration season, right? Those are very hard experiments done with birds, like 20 birds during migration seasons. They put birds inside cages. They want to see which direction the bird wants to migrate out of. Then they mess up with the magnetic fields and they see that the birds want to go out from another direction. There's a lot of evidence that is consistent with the behavior of birds, flies, actually being driven in a way, being driven by a spin model, like a spin-dependent chemical reaction acting under the hood. But there is nothing in between. It goes from molecules in solution to birds. So can we do better? And actually, we think we can. In my group, what we're doing is we want to bridge those two length scales there by actually developing glorified microscopes with coils. Okay, coils to impart different magnetic fields, and microscopes to start looking at those phenomena, say inside a single cell, for example. And when I tell you that we're building microscopes, our microscopes do not look like the things to your left from the internet. It looks like it's a big optical table. It's what you would expect to see in a atomic and molecular physics lab, a quantum computing lab, actually. And for a physicist, if there are physicists in the audience, I would say that what we're developing are experiments to study light matter interactions where matter is biomatter. But again, um, I don't care about words. Well, I do, but that's not as exciting as it gets. It turns out that there is, again, correlative evidence that the same type of spin-dependent chemical reaction might be behind many, many relevant biological phenomena, from the regulation of the production of of reactive oxygen species, which is like molecule, which are molecules that regulate a lot of signaling, and if you have too much of those like free radicals, it's bad for you. Cell growth is magnetic field dependent in a way that is consistent with them being driven by a spin model in planaria, in human cells. How much DNA repair gets done in bacteria seems to depend on magnetic fields in a way that is consistent with the spin model. But usually people really understand what I'm getting at when I show you the picture to the bottom of my slide there. And I'm going to take 1 minute to explain what this picture is. I have a friend who's a bona fide precision measurement physicist. He builds for a living very good Faraday cages, very good hypomagnetic chambers, which shield the interior from magnetic field. So the magnetic field of the Earth is 50 microtesla. Inside his chambers, they get a noise DC level of magnetic field of about 1 nanotesla. So what he did after hearing things about magnetic field dependence on doing things to biology, he grew tadpoles, which for 2 days, which was as long as he could go. He's a physicist without asking for a bio permit under two conditions. Once, and this has been repeated with controls by other people, the results are more or less the same macroscopically. In one condition, he put inside this hypomagnetic field chamber a magnetic field that is more or less like the Earth's. The embryos, the tadpoles embryos were normal. If, however, he left the embryos, new embryos, for 2 days only under this 1 nanotesla magnetic fields, about 30% of the embryos were macroscopically deformed. Okay, so here's what I just told you. You don't even need to apply a magnetic field, just taking out a tiny magnetic field and you create havoc in embryology. Okay, so again, nobody knows what's happening at the nanoscale. This is what we want to investigate. But given the strengths of the fields involved, everyone's best bet is that this involves spin-dependent chemical reactions. And this opens up a whole Pandora box from, like, if you want to colonize Mars, what's the magnetic field on Mars? How can we reproduce in Mars? Can we grow lettuce in Mars? To things more like down to earth. For example, can we learn how to deterministically tune those endogenous quantum degrees of freedom in biology to actually tune chemical reactions. Like you can imagine this as it being another way of doing medicine, an electromagnetic type of medicine. So the question that we want to answer is, can we establish or refute with those high-tech experiments that quantum physics is actually playing a big role on how biology works, right? We're not the only people thinking about this, there are other people. But our solution, and I think it's something very particular to our group, is that we want to throw absolutely high quantum tech onto, if you got curious about this, have a look at this, my little manifesto there on how to do responsible quantum biology. Those are the very, very brave people I have the immense privilege of working with, and I usually finish my talk by saying, may the quantum be with you. It was a pleasure to talk to you today.
Awesome. Thank you so much. Um, I do think we just have a few seconds. Actually, let me ask a quick question.
Yes.
Fran or Amir, are you on the line? Okay, awesome. Hi, Fran. Hi, Sounds Goody. Let me also.
So if you, I'll just answer the question. If someone wants to learn about quantum biology, there is a very good layman book that talks about the different flavors of quantum biology. It's called Life on the Edge by Jim O'Kalili and Joan Joe McFadden.
All right, yeah, awesome. Any of these other questions that catch your eye?
So, I mean, I just want to mention something, right? So right now, what I described, doing like electromagnetic field treatments, it's absolutely science fiction, right? But my goal is like to deterministically learn how each chemical reaction depends on each parameter of magnetic field intensity, frequency, so that we can learn deterministically how to tweak those chemical processes where I want to be in 30 years. Again, right now it's absolutely science fiction, right? But I mean, I want to be like, you have your phone, right? You say, well, today I need help with wound healing. Wound healing is something that depends on magnetic fields in a way that is consistent with the spin model under the hood. And then I go to my app and then I tap wound healing. And then your phone produces the exact magnetic field frequency and intensity that you need. And then you do some sort of remote medicine like this. Again, this is going to take time. I think it's time for us to start thinking about quantum mechanical degrees of freedom in biology by bringing experimental quantum physicists to the game, right? To have fun, because all experiments right now are done by biologists and chemists. Those experiments are beautiful, necessary, but I don't think they're sufficient. And I think that quantum engineers have a lot to contribute to this field on how to treat quantum mechanical degrees of freedom in biology as quantum things, right? So maybe in 20 years, we can have a quantum biology class like you're doing quantum computing now.
Awesome. Thank you so much for coming today and giving us this amazing talk. We really, really appreciate it. This is so fun. I hope everyone
Bye,
enjoyed it.
have fun, people. So proud of your efforts. Good work. Bye, friend. Bye, Dani. Bye, Sanskriti. Bye, bye. Take care. Thank you.
Thank you.